Okay, so I have this. There are a lot of perks to my job. I love my job, if you can't already tell. And there are a lot of perks, but I have to say the biggest perk of my job is the people that I get to work with. They are, and I'm not just talking about our patient advisors who are just excitable bunch, but our industry partners. I love it when we have regular meetings, and I know that I'm going to get to talk to the Janssen folks every 2 weeks, and I'm going to talk to the Zentis Bio folks, and I love it. Everybody is so kind, so appreciative, and so helpful that I feel like I have just the best job in the world. Another part of that, too, is I get to know people like Dr. Irina Murakhovskaya, because she is near and dear to my heart. I'm going to read your bio first so I don't cry right from the beginning. So, in case you can't read, Dr. Irina Murakhovskaya is an Associate Professor of Hematology. She completed a medical degree at Albert Einstein College of Medicine and residency and fellowship training at Montefiore Health System. Her clinical and research interests focus on autoimmune hematologic disorders, including autoimmune hemolytic anemia, paroxysmal nocturnal hemoglobinuria, and immune thrombocytopenic, I said that wrong, thrombocytopenic purpura. Thank you. As well as hematologic issues in pregnancy. Dr. Murakhovskaya is currently Chief of the Division of Hematology at Montefiore Medical Center and serves the wAIHA Warriors as Medical Advisory Board Director and Board of Directors Treasurer. Dr. Irina is also an avid chef, and she is currently on a Spanish kick. Dr. Irina has a fondness for truffles, margaritas, and day trading. Dr. Irina, day trading. Without Dr. Irina, there would not be a wAIHA Warriors. This group is the brainchild of Dr. Irina. Without her, we would not be here. And dare I say, many of us would still be, or I was going to say still ill, right? Or more ill. It is because of Dr. Irina that we are able to come together and be this amazing community that we are. Dr. Irina is also my friend, and I can't tell you, she has been a mentor to me. She's been a mentor to me. She's taught me the difference between an average margarita and a really good one. She told me how to do a Truffle of the Month Club. She is just an invaluable friend, and I can't tell you how much you mean to me. And we are thrilled that because last year we did not have a keynote speaker, we're thrilled that Dr. Irina is our first keynote speaker of the wAIHA Warriors annual patient meeting. Please.
Thank you so much. You're too kind.
Okay, smile.
Thank you. Okay, yeah, pressure. Okay, so I'll stand here so I can see everything. And this is going to be very basic, so I'm sorry if you guys expected something finer. But yeah, so I'll just do a brief introduction on genes, genetic changes, next generation sequencing, and what it means for autoimmune disorders and warm autoimmune hemolytic anemia. So, and these are not genes that we're going to talk about. We're going to talk about the genes that make up our genetic information, and we're going to talk about genes and DNA. So all of the living things, including plants, right, are made up of cells that carry DNA. The cells in general consist of nucleus, which carry the genetic material, and then the cytoplasm, which carries machinery that processes this genetic material and makes things. So DNA, or the fancy term is deoxyribonucleic acid, and I practiced it today, is a molecule that contains really a genetic code, so kind of like a computer code, that programs the organism's makeup. And it's determined by the order of 4 molecules, or nucleotides, the fancy name for it, the specific molecules. They're called adenine, thymine, cytidine, and guanine, so ACTG. And that's basically the way, the order of these molecules is it kind of programs how the genes are constructed. So the genes, individual genes, are made up of these DNA molecules. So this is the DNA, a chunk of DNA is a gene, and then multiple genes comprise a chromosome. And in the nucleus of each cell, the DNA is basically packaged in a chromosome, and this DNA is really tightly wound up around proteins that stabilize it and support it. And those proteins are called histones. So you can see it really just like DNA is like a thread that's wound around the histones. And as I've mentioned, each chromosome contains multiple genes. And the reason 23andMe is called 23andMe is because each of us has 23 pairs of chromosomes. We get one set from a father, get one set from a mother. So 46 chromosomes altogether. And on the right here, you can see what they actually look like. So if you take a picture and line them up, this is a chromosome 1, this is a chromosome 2, etc. The very last one is the X chromosome, and this happens to be a female, so they have 2. So there's no Y chromosome. But if it was a male, there would be a teeny beeny Y chromosome instead of the extra X one. Okay. So then what happens next? So we take this genetic information from the DNA and we rewrite it, or the fancy term for that is transcribe it, into a message that we send out from the nucleus into the cytoplasm. And that message is called messenger RNA, which is a molecule similar to DNA, but a little different because it can leave the nucleus and go into the cytoplasm. And then we use the genetic code contained in this messenger RNA to translate this code into making proteins. And that process is called translation. So basically those two processes of transcription, right, creating a message that leaves the nucleus, goes into the cytoplasm, and translation, using that message to change it into a protein structure, we make different proteins. And that's basically how, how the biology of us works, right? So you have this machinery that reads your DNA and makes specific protein. And that will tell you whether you're going to have blue eyes or black eyes or, blue hair, brown hair, big nose, small nose, and, or predisposition to an autoimmune disease, right? But if we say that everybody carries the same DNA, right, why are we having different cells? We have the lung cells, and we have the hair cells, and we have the nose cells, even though we are making them from the same DNA. And the reason this happens is because not all the genes are active or transcribed in every single cell. So we selectively transcribe different genes in different cells. And this selective transcription is really responsible on the path the cells will take, whether they become cells that line your lungs or the cells that, make a uterus, or the cells that produce your hair, or your blood cells. So different genes, different tissues express different set of genes that makes them different. So how do these tissues know what genes to express? So there are actually proteins that are responsible for regulation of the expression of the genes. And they're kind of like a on and off switch, right? So you can turn on the gene expression or you can turn it off and tell the cell, we're not touching this gene here. This protein we're not going to make in this cell. And a lot of these controlling genes have to do with epigenetic changes. So that fancy term refers to changes that are not affecting your DNA itself. They just basically put a little molecule on top of, or a little modification on top of the DNA, and makes it difficult for the machinery that transcribes the DNA to attach to a DNA. So therefore, you can't attach, you can't make the protein, the gene is silent. Or the other way around, you put a modification on top of a DNA that makes you more likely to have attachment of proteins that read the DNA and transcribes the protein. So epigenetics basically refers to not changes in expression of the genes without actually changing your DNA, just by modulating ability of DNA transcribing machinery to attach to the DNA. And they look really like little molecules like this. So this is your DNA, and then there's a little molecule that sticks here, and let's say it doesn't allow the proteins to bind. And they can happen on a DNA. They can also happen on those proteins that I mentioned before called histones. And interestingly, this is the one thing that can actually, the epigenetics, right, the attachment of these molecules, methyl groups typically, or acetyl groups. This is the one thing that can be influenced by non-genetic things, so like your diet, your age, your lifestyle, and this is actually something you can do something about, right? And errors in these epigenetic processes, such as, you don't modify the right gene or you modify the wrong gene, basically leads to abnormal activity or inactivity of the gene, and that can lead to a disease as well. So now we know all about genes. I'm going to talk about the genetic sequencing. So the genetic sequencing is basically the process of determining the precise order of these nucleotides in a DNA molecule, and allows the scientists to basically read this code, like the computer code, right, of the DNA, and provides the information about the makeup of the genetic makeup of the individual. It wasn't developed that long ago. It was 1977, less than 50 years ago, that we first discovered the genetic sequencing. And it was done by Dr. Sanger. And at that time, it was a very difficult process. It was very labor-intensive, very expensive. It took a long time, which is why it took us so long to get to this process, to get to this point. Now we have what's called next-generation sequencing. And basically, it's just more sophisticated testing techniques that allow us to look at these genes faster, more cost-efficient, cheaper, and we can process large amount of material in a relatively short period of time, and it costs less money. So we can actually finally start sequencing people and their DNA. Like 23andMe. So what are the applications of the genetic sequencing? How is it useful in everyday life? For example, forensic genomics, right? You can identify individuals, for example, criminals or victims using their DNA, right? You hear in the news that the victim was identified using DNA. So that's one of the ways. You can solve criminal cases. You can establish who the father of the baby is, right? Based on their, comparing whether his genes are similar to the baby's. It's been instrumental in the diseases like COVID, right? In microbiology, we can sequence the microbacterial genomes and figure out evolution and pathogenesis and resistance to antibiotics of different bugs, but also identify new viruses and outbreaks and trace them and figure out, well, this is a new variant of COVID, right? And this variant is called this, and this called that, and that has to do with different genes that they have, because COVID has genes too, right? It helps us in agriculture with crop improvement, genetically modified organism development, which I'm not a fan of, but also ensures the food safety and security, and then lots of other things like archaeology and biotechnology and environmental studies. So how can we apply the genetic sequencing in medicine? There's a number of ways we can apply it. We can use it for diagnosis, and I'll go over this a little bit more. We can identify the people at risk for the disease. We can use it to prognosticate, and we can use it to guide treatment. So for diagnosis, there are lots of diseases that are what's called monogenetic diseases, meaning you have the gene, you're gonna have the disease a lot of times, right? And presence of the gene develops disease. And you guys heard of those. Cystic fibrosis, Gaucher's disease, Huntington disease, hemophilia, Down syndrome, sickle cell disease, some immune deficiencies, and some hereditary cancer syndromes like BRCA1, BRCA2, Angelina Jolie, right? I mean, you guys heard of that. We can also diagnose some acquired diseases. So some of the mutations are associated with cancer development, progression, right? And we can also identify new disease-causing mutations that we haven't even heard about, right? We can also identify people at risk and potentially use it for prevention. So an example would be there is a condition called hemochromatosis, where you carry a mutation. It's a Viking disease. It developed presumably a long time ago in Vikings to prevent them from dying from bleeding out. And basically, you have an increased predisposition to iron accumulation. So you absorb all the iron you can get. And these people get extreme iron overload that can lead to cirrhosis and can lead to liver cancer. And the treatment is very easy. If you pick them up early enough, you just turn them into blood donors, and you bleed them every couple of months, and it's good for everybody, and they don't get sick, and we have, continuous blood supply. I have a few of those. It can also actually help you to predict risk of side effects of medication. So certain chemotherapeutic agents, for example, have higher chance of toxicity in people with certain genetic mutations. So you can check for those mutations and see if you need to adjust the dose. And the same applies to, obviously, non-chemotherapeutic things, but I just don't know them as well. You can also use it for prenatal diagnosis, right? So if you know that the mother is a hemophilia carrier, for example, and so you can use it to see if the baby has hemophilia or not. And you can identify disease even before the baby is born. And if you think far enough in advance, right, you can even use it for preconception counseling. So this is quite helpful. You can also use it for prognosis, as I've mentioned. So for example, in leukemias and lymphomas, certain genetic mutations are associated with worse prognosis or better prognosis. And this actually guides our treatment decisions. So if we know that this is a condition that's very likely to relapse, very bad cancer, we intensify the chemotherapy. And if we know that this is, relatively easy to treat disease, then we actually go easier, less chemotherapy, less cycles. We can also use these things to track the response to treatment, right? So we can check this, it's one of the markers, right? So we check the blood and see if the person is responding to whatever treatment we're giving. We can also use it to predict the risk of recurrence, and again, decide if a more intensive treatment is needed. Certain genetic mutations, congenital or acquired, are associated with certain diseases, like cardiovascular disease and heart failure, or autoimmune disorders, such as rheumatoid arthritis and lupus, or inflammatory disorders, such as inflammatory bowel disease and COPD. And then for treatment purposes, we can use these genetic results to personalize treatment, to develop targeted therapies. If we know a mutation the person has, we can potentially to determine a treatment that targets this specific mutation. And hopefully, because it's a targeted treatment rather than, a hammer, we can minimize the side effects of treatment. And again, we can monitor response to treatment and monitor side effects. And we can use it for gene therapy. And you guys probably heard that there are a number of gene therapies in hematology that have been approved recently: sickle cell disease, hemophilia, thalassemia. And this is when we can actually, this is your DNA, and we can actually cut out that bad gene and then put in a good one, and then put it back in the person. And that's called gene editing, right, when we edit the gene. So it's quite amazing what eventually we'll be able to do. So what about the genetic sequencing and autoimmune hemolytic anemia? So I'm going to talk a few minutes about the pathogenesis of autoimmune hemolytic anemia. And I know this looks a little complicated, but basically, you have a combination of certain genetic factors, and this probably applies to every disease. You have a combination of certain genetic factors, immune dysregulation, and environmental factors that lead to what we call breakdown of immune tolerance. So your body is not tolerating your own cells. You're developing cells that attack your own body when they're not supposed to. And we know of certain associations with warm autoimmune hemolytic anemia. So, and as many of you know, there are congenital syndromes and immune deficiencies, most common being CVID, or common variable immune deficiency, that has a higher prevalence in patients with warm autoimmune hemolytic anemia. And a little bit less common, but still a very relevant one, is ALPS, or autoimmune lymphoproliferative syndrome. That's also a genetic condition that can be seen more commonly in patients with warm autoimmune hemolytic anemia. But also other things like IgA deficiency and Kabuki syndrome, which is associated with a very particular mutation that I'm going to talk about a little bit later. And then we all know that autoimmune diseases like lupus, inflammatory bowel disease, and sarcoid, as well as rheumatoid arthritis, have a higher prevalence in people with warm autoimmune hemolytic anemia. And there are association between acquired things like viral infections, lymphoproliferative disorders, solid tumors, and more recently, we actually use medications to alter the immune system in some cancers and, and engage immune system in cancer fighting, and that causes autoimmune side effects. Introducing somebody else's immune system or somebody else's tissue, like transplant, also messes up with your immune tolerance and can cause autoimmune hemolytic anemia. And we know that there is an increased incidence of inborn errors of immunity in patients with warm autoimmune hemolytic anemia. And some clues that that may happen include patients who have a younger age of diagnosis, who have very difficult relapsing refractory course, who have multiple problems with their immune system, including multiple blood count issues. We call it Evans if you have more than one cytopenia, or multiple autoimmunities, right? So you have multiple autoimmune conditions. And then there are certain immunologic blood test results that we'll look at as well. And these are usually diagnosed by next generation sequencing, or sequencing the entire genome, or exome rather, but genome. So there was actually a few, I wanted to just review a couple of studies that have been done to look into that in people with autoimmune cytopenias, and that's pretty much going to be, and let you rest the rest of the time. So this was a study looking at children, because we do know that younger age of diagnosis is associated with higher tendency to have a genetic problem. And the study took 47 patients with autoimmune cytopenias, including ITP, that's immune thrombocytopenic purpura, as well as autoimmune hemolytic anemia and autoimmune neutropenia, which is when you attack your white cells as opposed to the red cells, and ITP is when you attack your platelets. And those were children age 0 to 18 at the onset of the disease. And when they were sequenced, inborn errors of the immunity, so those are the genes that regulate your immune system, that we know that regulate your immune system, were present in 19 of 47 patients. So this is known genes that we already know. So almost half had something that was genetically predispose them, including 9 with common variable immune deficiency. So that's the most common one. But then also 4 with autoimmune lymphoproliferative syndrome, and then some other conditions. And it's usually associated with low lymphocyte counts as well as other immune abnormalities. And we actually know that presence of some inborn errors of immunity can already guide your treatment. So there are some conditions that will respond better. For example, let's say those stat things. And by the way, the mutations, whenever you see the mutations are usually the 4-letter words. Just that, they're usually designated by, a combination of 4 letters. So there are some mutations, STAT3, right, the 4-letter word and a number, gain of function mutations. So these mutations respond better to certain treatments because of the way, the pathophysiology works, because they activate a specific pathway in the body. And we actually can now already specifically target that particular pathway. For example, patients with autoimmune lymphoproliferative syndrome, right, ALPS, respond to sirolimus, right? And that's a medication that some of you have heard of and we already have available. There are some patients who have mutation that's called PI3K kinase mutation, that respond well to PI3K kinase inhibitors that we already have on the market. And there are some patients that we know if this is the mutation they have, they will need to be, in order to be cured, they would need a stem cell transplant. And so in Evans syndrome, which many of you guys know what it is, but it's basically presence of 2 autoimmune cytopenias. Usually it's warm autoimmune hemolytic anemia and ITP. I believe it was the French group that looked into presence of mutations that potentially have something to do with your immune system. And they found these mutations in 65% of children. When they looked at the adults, and this was as recent as this December, they presented the data, they saw only about 11% had the genetic diagnosis. So probably less common in adults, or maybe we're not looking in the right place, right? Okay. Cold agglutinin disease, right? Some of you guys have cold agglutinin disease. So there is recurrent mutations in KMT2 gene, right? KMT2D gene, right? The 4-letter word, that were seen in 11 out of 16 patients. And another gene, the CARD11 one, that was seen in 5 out of 16 patients. So basically 16 patients had, out of 16 patients who had this genetic sequencing performed, all of the 16 had some kind of recurrent mutations. And it's important because there are drugs that we already have on the market that may potentially have efficacy in this condition. We haven't used them yet. There is no clinical trials in this direction, but there are drugs that can potentially target that pathway. The KMT2 is actually one of those epigenetic things. In ITP, if you look at, which is immune thrombocytopenic purpura, is when we destroy the platelets as opposed to the red cells. 37%, and this was actually done by our own Dr. Bruno Fatizzo and presented again this year's ASH meeting. He looked at ITP patients, and 37 patients with ITP he did the sequencing on, and out of them, 40%, 38% had at least one mutation on next generation sequencing. Now in his study, he actually excluded mutations that were likely to be genetic. And the point of this is that you probably can acquire these things as well. And to some of your points, yes, TET2 was a very common mutation, but you could see, which was seen in about half the cases. But the other mutations were seen in a number of people as well. And these are mutations that you get as you age, basically. And they do predispose you to a few things, like, for example, the DNMT3A predisposes you to heart disease, as well as bone marrow problems, as well as possibly autoimmune cytopenias. And then we actually looked at particular mutations in autoimmune cytopenias, and our idea was, well, if the patients with cold agglutinin syndrome, majority of them have this KMT2D mutation, why don't we look at other patients with autoimmune cytopenias and see if they have this KMT2D mutation? And we actually looked at patients with all of the autoimmune cytopenias, predominantly ITP, but also warm autoimmune hemolytic anemia, and we had a couple of cold autoimmune hemolytic anemia patients. And out of 55 patients with autoimmune cytopenias, 20 had these mutations. So it was a particular set of mutations. They were called KMT2C and KMT2D. They're epigenetic regulators that have to do with gene silencing. So if you basically have a mutation in this gene, you don't silence your genes, certain genes are out of control, and that may predispose you to autoimmunity. The caveat, and then regarding the warm autoimmune hemolytic anemia, we had 12 patients, and out of them, almost half had presence of these mutations. So this is very early. The caveat to that is our population is mostly Hispanic. 12 out of 20 patients with these mutations were Hispanic, so it could be that this is something that's present in the Hispanic population that predisposes them to autoimmune disease. And the mutations that we saw in the warm autoimmune hemolytic anemia, 2 of them had the KMT2C, 2 had KMT2D, and 1 had both. And it didn't seem like it affected, the one that had both, it didn't seem like it affected what they had. And that's all I have for you. Hopefully. Okay, questions? Yes?
So you talk about genetic sequencing and the research that's been done. What if we as patients are interested in getting genetic sequencing? Where do we go? How do we do that?
So,
Sure. So my question is,
no, he wants me, so because, so the question is, I discussed the genetic sequencing, and so how do patients get the genetic sequencing? So currently, this is not a standard of care, right? So this is not approved for routine treatment. It's still investigational. Therefore, the insurance companies are unlikely to pay for this. This is the sequencing can be done on the bone marrow, but this also a blood test. This could be done as simple as a blood test, the same tube that you do your CBC. If your doctor says that you have blood, if you have a cytopenia, low blood count, which is unexplained, then the insurance will cover it. So it will, so that's, and pretty much any hematologist can send the sequencing. We send it routinely for people with malignancies. Now, there are genetic sequencing specific for inborn errors of metabolism, like I've mentioned, these, the ones that we see in kids. And that's a genetic test. Again, either your hematologist can send it, sometimes immunologists are a little bit better with it. And it's just an immunodeficiency panel, and a lot of labs do it. And that's kosher to do with autoimmune disorders. Yes?
Dr. M, that was a fantastic talk. I do want to piggyback off of what you said, just to clarify that we send some of these genetic tests as well. I do want to emphasize that it's not a catch-all. Even
Right.
the best genetic tests catch about 700 or so mutations out of the billions and billions of genes that you have.
Right.
And so it's, if you have it, you have some information, but if you don't have it,
It doesn't mean that it's not there. Absolutely, absolutely. So we're still learning, right? This is a very new field. Like I said, remember that the very first sequencing was done less than 50 years ago. So, yes?
You
So the stuff that I'm talking about, you can. So you need to compare the mutations that you see in the blood to mutations that you see, let's say, like in a buccal swab. So like, we can get some DNA from your cheek cells, from inside, and then look and see if the same mutation is present. So that's one of the way to tell. The other way to give us an idea is to see what's the percentage of abnormal cells that we see, and we do get that information. So if we see about 50% of the cells carry the mutation, then we assume it's genetic. But again, to confirm, you need to compare it to other cells to make sure it's not just the blood cells that carry it. So, yeah. Mm-hmm. I guess eventually, yeah. Eventually, right? We're early. Yeah. So, but it's done. It's done in some conditions already, but for BRCA, it hasn't been done yet, yeah. Yeah, I mean, there is a caveat to that. So in order to do this gene therapy, the process is basically they take your cells, right? And it's usually hematopoietic cells, so I'm not sure if every cell in the body can be changed, right? So they take your cells, then they do whatever they do with the gene editing, right? And then they put it back in the body, but before they put it back in the body, they give you a strong chemotherapy that has a lot of side effects. So we're not quite there yet to make it easy peasy, fix the genes and stuff like that. It's not that simple. It's kind of like almost as intense, not quite, but almost as intense as a transplant.
And I can speak a little bit to that because I work in oncology, and what you're talking about is called CAR-T, chimeric antigen receptor therapy, and it's not something that you want to do because it has a high incidence of neurotoxicities where people become psychotic from the treatment itself. So it's not a walk in the park.
Yeah, it's getting better. Yeah. So it's better than having sickle cell disease, right? But for a person who, can get away with a, surgery, which is, not the best thing, but it's a much easier treatment, than having the gene therapy. Yeah. Whoever takes it. No. Yeah, I mean, the research, yes, we do publish this. We're working on this paper right now. Yeah, I mean, we publish it in hematologic journals. Yeah. But it's not easy. Okay, any other questions? Okay, all right, thank you.